Penicillin lasted less than 20 years before bacteria figured out how to beat it. Methicillin lasted 2. Vancomycin, once called the antibiotic of last resort, began failing within a decade of widespread use. Carbapenems, the drugs hospitals reach for when nothing else works, now face resistance that kills 216,000 people every year. Every single synthetic antibiotic ever created has eventually been outsmarted by the organisms it was designed to kill. But there is a plant that has been fighting the exact same bacteria for over 4,000 years. The same species, the same strains, the same pathogens that now shrug off our most powerful pharmaceutical weapons. And in 4,000 years of continuous use across 4 continents, bacteria have never developed resistance to it. Not once, not partially, not in any documented study in the history of botanical medicine. In Germany, doctors can prescribe plant-based medicines. In China, this plant remains a mainstream treatment. Across Africa, South America, and Southeast Asia, traditional healers never stop using it. In the United States, it is classified as a weed. Agricultural extension offices publish guides on how to kill it. Herbicide companies sell products specifically formulated to eliminate it from your lawn. The plant is called Biden's pilosa, and the reason bacteria cannot beat it is the same reason the American pharmaceutical system will never sell it to you. In 1928, Alexander Fleming discovered penicillin. By 1945, he stood before the Nobel Prize Committee and warned the world that bacteria would eventually learn to defeat it. He was ignored. Doctors overprescribed. Farmers pumped antibiotics into livestock. Pharmaceutical companies marketed broader and broader spectrum drugs because broader meant more profitable. By 2021, more than 1 million people were dying each year from infections that antibiotics could no longer cure. Deaths from methicillin-resistant Staphylococcus aureus alone more than doubled between 1990 and 2021, rising from 57,000 to 130,000 annually. The Lancet now projects that between 2025 and 2050, antibiotic-resistant infections will directly kill 39 million people. That is 3 deaths every single minute for 25 years. South Asia alone, including India, Pakistan, and Bangladesh, faces 11.8 million of those deaths. Here is what nobody is telling you about why this is happening. It is not just that bacteria are becoming resistant. It is that our entire strategy for fighting them was flawed from the beginning. A synthetic antibiotic is a single chemical compound designed to attack bacteria through one specific mechanism. Penicillin disrupts cell wall synthesis. That is it. One target, one lock, one key. When you send a single mechanism weapon against an organism that reproduces every 20 minutes and mutates with every generation, you are not fighting a war. You are running a countdown. Bacteria defeat single compound antibiotics through straightforward evolutionary math. A population of 1 billion bacteria contains roughly 1 million random mutations. If just one of those mutations alters the target site the antibiotic attacks, that bacterium survives while every competitor dies. Within hours, the resistant strain dominates. Within days, it is the only strain left. Some bacteria do not even need to mutate. They share resistance genes horizontally, passing genetic instructions between unrelated species, like passing notes in a classroom. A resistance mechanism that evolves in one bacterium can be borrowed by a completely different pathogen within hours. This is why every antibiotic fails, not because the drugs are poorly designed, because the strategy of one compound against one target gives bacteria a problem simple enough to solve. Now consider what Biden's pilosa does. Scientists have identified more than 200 bioactive compounds in this single plant. 70 aliphatics, 60 flavonoids, 25 terpenoids, 19 phenylpropanoids, 13 aromatic compounds, 8 pterins, multiple polyacetylines with unique photosensitive properties. These compounds do not attack bacteria through one mechanism. They attack through dozens simultaneously. The flavonoids and phenolic compounds rupture bacterial cell membranes, causing them to leak and collapse. The terpenoids penetrate cell walls and disrupt the respiratory chain bacteria need for energy production. Other compounds interfere with quorum sensing, the communication system bacteria use to coordinate attacks and form protective biofilms. When bacteria cannot communicate, they cannot organize the collective behavior that makes infections lethal. Still, other compounds bind directly to bacterial DNA and prevent replication. Others block protein synthesis. Others inhibit the efflux pumps that bacteria use to expel antibiotics from their cells. The polyacetylenes absorb ultraviolet light and become phototoxic, growing more potent when exposed to sunlight, killing bacteria and yeast on contact. For a bacterium to develop resistance to Biden's pilosa, it would need to simultaneously evolve defenses against all of these mechanisms at once. Not one mutation, not 10, dozens of independent mutations, all occurring in the same organism, all conferring survival advantages, all at the same time. Even if partial resistance emerged against a few compounds, the remaining mechanisms would still kill the bacterium. Penicillin gives bacteria a puzzle with one piece. Biden's pilosa gives them a puzzle with 200 pieces that all need to be solved simultaneously. In 4,000 years, bacteria have never solved it. In 2014, researchers in Brazil tested Biden's pilosa leaf extracts against 60 strains of oxacillin-resistant Staphylococcus aureus. These are hospital superbugs, the bacteria that survive surgery only to kill patients through wound infections weeks later. The leaf extracts inhibited growth of every single strain tested. The mean inhibition zone was significantly higher than chlorhexidine at 0.12%, the gold standard antiseptic used in hospitals worldwide. The extracts also showed no toxicity at the concentrations used. A plant extract outperformed pharmaceutical-grade disinfectant against bacteria responsible for 130,000 deaths per year. Other studies followed. In 2018, researchers tested Biden's pilosa against waterborne bacteria that cause diarrheal disease, one of the leading killers in developing nations. Every bacterial species tested was sensitive. The dichloromethane extracts showed particularly strong activity with an average minimum inhibitory concentration of 0.56 milligrams per milliliter. These are concentrations achievable in real-world applications. The plant extracts also showed no toxic effects on muscle cell lines, meaning the concentrations that kill bacteria do not damage human tissue. A 2021 study examined the plant against sexually transmitted infection bacteria. The results showed significant activity against Neisseria gonorrhea, the bacterium that causes gonorrhea, and Gardnerella vaginalis. Multiple extracts were also effective against Candida albicans, the fungus behind persistent yeast infections that afflict millions of women worldwide. A 2017 study showed that Biden's pilosa extract produced an 18.2 millimeter inhibition zone against E. coli at 10 milligrams per milliliter, competitive with pharmaceutical-grade ampicillin. A 2016 study demonstrated that the plant extract could preserve ground beef more effectively than BHT, the synthetic preservative used in commercial food production, lowering total viable bacteria counts over 6 days of cold storage. The antibacterial activity was not limited to human medicine. It extended into food safety, veterinary applications, and aquaculture, where a 2022 study confirmed strong activity against pathogens devastating fish farms. Study after study confirms the same pattern. Biden's pilosa demonstrates broad-spectrum antimicrobial activity against Pseudomonas aeruginosa, Klebsiella pneumoniae, Bacillus subtilis, and Escherichia coli. The bacteria behind urinary tract infections, wound infections, pneumonia, and meningitis. The infections filling emergency rooms. The infections that antibiotics are increasingly failing to treat. The evidence is overwhelming. The plant works. Traditional medicine was right. So why does the regulatory response to this evidence differ so dramatically depending on which side of the Atlantic you are standing on. In 1978, the German government assembled a panel of physicians, pharmacologists, toxicologists, and scientists and gave them a single task: evaluate the safety and efficacy of every medicinal plant sold in German pharmacies. Not as supplements, not as wellness products, as medicine. The panel was called Commission E. Over 16 years, it reviewed more than 380 plants, publishing official monographs with approved uses, dosages, contraindications, side effects, and drug interactions. The panel included physicians who prescribe plant medicines, pharmacists who dispense them, and scientists who studied their chemistry. They assessed safety on a doctrine of absolute certainty and efficacy on a standard of reasonable certainty. When they finished, they had created the most comprehensive official evaluation of herbal medicines in the Western world. German doctors began routinely prescribing plant-based medicines alongside synthetic drugs. German pharmacies stocked them on the same shelves. A large proportion were sold on prescription. Germany's herbal medicine market grew to represent 10% of all pharmaceutical sales. The European Union formalized this approach in 2004 with the Traditional Herbal Medicinal Products Directive, creating a simplified registration pathway for plants with at least 30 years of documented safe use. Plants do not need to pass the same clinical trial gauntlet as novel synthetic drugs. They need to demonstrate a tradition of safe use and a plausible mechanism of action. France restricted licensed herbal products to pharmacy-only sales, treating them as medicines requiring professional guidance. In the United States, the FDA took a different path entirely. You can sell a plant as a dietary supplement with no therapeutic claims, or you can spend hundreds of millions of dollars pushing it through the same approval pipeline designed for single-molecule synthetic drugs. Phase 1, Phase 2, Phase 3. Identical to penicillin. There is no middle path. There is no simplified registration for plants with millennia of documented safe use. As of today, only 2 botanical drug products have ever received full FDA prescription approval. 2. In the entire history of American pharmaceutical regulation. The FDA evaluates drugs as single active ingredients with measurable dose-response curves. Biden's Pelosa contains more than 200 compounds working synergistically. Isolate a single compound and you lose the multi-target advantage that makes the plant work. Keep the whole extract and you cannot standardize it to the specifications the approval process demands. Patent protection for whole plant extracts remains unclear. Investor pessimism is chronic. The process is capital-intensive, and any loss of funding kills the project. The regulatory system was built for penicillin. It was never rebuilt for Biden's Pelosa. Meanwhile, the antibiotic pipeline is collapsing under the weight of the same economics. The WHO's latest review found only 27 antibiotics in clinical development targeting priority pathogens. Only 6 were classified as innovative. Pharmaceutical companies abandoned antibiotic development decades ago because chronic disease medications taken daily for life generate exponentially more revenue than antibiotics prescribed for 7 to 14 days. Between 1999 and 2014, fewer than half of new antibiotics that reached market were available in more than 10 countries, and those were predominantly wealthy nations. The developing world, where resistant infections kill the most people, receives new drugs last and loses access to old ones first. 80% of the world's countries have extremely limited access to effective first-line antibiotics. More people now die from lack of access to antibiotics than from drug-resistant infections themselves. And this access gap accelerates resistance because undertreated infections become breeding grounds for resistant strains. And yet half the world never stopped using Biden's Pelosa. In China, healers have prescribed it since at least the 7th century. They called it Xian Feng Chao and used it for enteritis, bacterial dysentery, pharyngitis, and infected wounds. Traditional Chinese medical texts describe it specifically as a remedy for clearing heat and removing toxins from the body. In Japan, it became the primary ingredient in traditional compresses for inflammatory skin conditions, a practice that continues today. When Spanish colonizers arrived in the Americas in the 16th century, they documented something remarkable. Indigenous peoples across Mexico, Central America, and South America were treating wounds that would not heal, stomach infections, dysentery, and fever, with a plant they had cultivated for generations. The colonizers recorded it under various names. The indigenous peoples had their own names, passed down through centuries of continuous medical use. In Uganda, the Banyankore people applied fresh leaves directly to open wounds. South African healers prescribed it for headaches, kidney problems, malaria, and as an antidote to certain poisons. In Vietnam, soldiers adopted it as both food and battlefield medicine during the war, earning it the name soldier vegetable. In Kenya, it was called michicha, and eaten as a nutritious green valued for both food and healing. In India, it treats wounds, colds, hepatitis, glandular sclerosis, and urinary tract infections. The Food and Agriculture Organization of the United Nations has recognized it as an edible plant since 1975. 4 continents, dozens of independent cultures. None had contact with each other when they discovered that this specific plant fought infection. Traditional healers applied fresh leaves to wounds and left them exposed to sunlight. They did not know that the polyacetylenes inside the leaves were absorbing ultraviolet light and becoming phototoxic. They did not know the photochemistry. They knew that it worked. 4,000 years of empirical observation confirmed by 21st century laboratory science. Biden's pilosa is an annual herb growing 30 to 100 centimeters tall. Pinnately compound leaves with serrated edges. Small yellow flowers with white ray florets. Black seeds armed with barbed bristles that hook onto clothing and animal fur. One of nature's most efficient dispersal systems. It grows in disturbed soil, vacant lots, agricultural margins, driveways, and the cracks in sidewalks on every inhabited continent. It produces hundreds of seeds per plant. It tolerates a wide range of conditions. It is extraordinarily difficult to eliminate once established. It costs nothing. It requires no cultivation. It requires no pharmaceutical manufacturing infrastructure. The very characteristics that make it a successful weed make it an accessible medicine. It grows where people live. The Lancet's modeling shows that improving access to antimicrobials, including plant-based alternatives, could prevent 92 million deaths between 2025 and 2050. New antibiotics targeting gram-negative bacteria could avert another 11 million deaths, but those drugs need to be developed, funded, approved, manufactured, and distributed to the countries where people are actually dying. Biden's pilosa requires none of that infrastructure. It is already there, growing in the soil, ready to harvest. In Germany, this plant could become a prescription medicine. In China, it already is one. Across Africa, Asia, and Latin America, it never stopped being one. In America, it is a weed growing through the cracks of a hospital parking lot. We built an entire medical system around single compound, single target drugs, and then acted surprised when single-celled organisms found the one mutation needed to survive. We built a regulatory system that demands plant medicines behave like synthetic molecules, and then acted surprised when no one could afford to prove they do. Meanwhile, the plant that fights bacteria through 200 simultaneous mechanisms, that has never lost effectiveness in 4,000 years of continuous use, that outperforms hospital-grade disinfectants in peer-reviewed studies, grows within walking distance of almost every human being on Earth. It is called Biden's pilosa. Bacteria have never beaten it, and 3 people die from antibiotic-resistant infections every minute while regulators decide what category to put it in.